Okay, hello everyone, welcome back. So our first speaker of today is Hideshi Ooka. Be telling us about timescale decomposition of CRNs and implications towards autocatalysis. Thank you, Hideshi.
Yeah. So, Praful, thanks for the invitation, and thanks for inviting me to be not just a speaker, but since last semester, inviting me to join in this seminar series. I've learned a lot so far, and I'm quite excited to give the talk today. One disclaimer is that even though I say timescale decomposition, it's not like a really fancy technique. I mean, it's just like a decomposing the linear matrices into one matrix into two so that it's easier to calculate. And but I'm hoping my background is probably very different from everybody else. And so I thought maybe it would be interesting to see how that kind of technique can be applied to different problems. And so in the first part, I am going to talk about artificial catalysis, like industrial catalysis. And then in the second part, and that's where I kind of developed the technique of the timescale decomposition. And then in the last half, in the second half, I will talk about it being applied to autocatalysis and in the context of origin of life. So this is kind of my background and overview of what I'm doing. So actually, my main research topic is not autocatalysis. It's like not non-autocatalytic catalysis. And what I mean is, for example, one of my main topics is to make hydrogen from water. So water electrolysis towards sustainable hydrogen production. And another of my topics is, for example, designing better enzymes for biocatalytic reactors. And the kind of overview, even though those physical systems look different, of course, their theoretical aspects are very similar because when you think about them in terms of mass action kinetics, they're quite similar. I mean, they are both catalytic. And also, I kind of have this overview of the cycle between water and oxygen that has been kind of governing the energy material conversion on Earth for a very long time. What I mean is, right now we are breathing oxygen, and we use oxygen, and we burn for example, sugar or some kind of chemical fuels, and after that goes back to water, and this is how we get energy. But where that carbon molecules come from, where those reduced carbon molecules come from, they come from photosynthesis. So the first order generation of chemical fuels in the natural environment today, it comes from photosynthesis. And then, for example, maybe trees makes leaves, and those leaves are eaten by some animals, and then we eat those animals. So the food chain, it's supported by photosynthesis. So it's based, and the photosynthesis, if you recall your biology courses, then it uses CO2, it uses sunlight, and it uses water. So this water is used as the reducing agent to make those chemical fuels. And in my eyes, that's exactly what people in industry are trying to do, the chemical engineers are trying to do in terms of the hydrogen economy. So what that means is the people are trying to start from water and give some energy to water, and that would liberate some hydrogen. So this is the chemical fuel, and in that process we get oxygen. That's a how to say leftover byproduct because you strip off the hydrogen, so then you get left with the oxygen, and then in the fuel cell you can burn the hydrogen, combine it with oxygen, and you get water again. And so this kind of cycle between water and oxygen, this is the energy material conversion that is happening in nature. And this is kind of what the chemical engineers and those technology people, what we are aiming for in terms of increasing the sustainability of our society. And as you might guess, we need catalysts to do that. And in terms of catalysts, it makes sense to think about what kind of device we are actually using them in, what kind of reaction environment they are in. And in the case of, for example, generating the hydrogen, which is one of the main projects I'm going on, one of the main projects I'm focusing on, one of the main devices is called a PEM. That stands for a polymer electrolyte membrane. And what this electrolyzer does is you pump water in from one end, and then it spits out oxygen and hydrogen from the two different electrodes. And these are the catalytic regions, except the problem is it's super acidic. And the reason is because you need some kind of electrical conduction here, and the reason in conductive, how to say, the carrier, charge carrier in this case is a proton, and that's, as you know, a proton is acidic, so it has a really acidic environment in this reactor chamber, and that's why it has a high conductivity and high reactivity and high efficiencies, except it's super corrosive to almost all of the materials that we have so far. And that's why in industry we use a material called iridium oxide. And iridium is actually found here. It's right next to platinum. And so you guys know platinum is expensive, and iridium is even more expensive. Not only is it expensive, it is a byproduct of platinum, meaning, so if you have a lot of money, then you can buy more platinum. And if you have a lot of money, then you can ask someone to dig more platinum. But even if you have a ton of money, you can't ask someone to dig more iridium. It comes as a byproduct of platinum. So a huge problem in this field is how can we make catalysts which are alternative materials to this super rare metal iridium? Can we make something that's as good as iridium using some different metal? And one of the approaches my collaborators and I are working on is making inorganic materials using, for example, manganese. Manganese is right next to iron. So manganese is this pink one here. It's right next to iron. And so it is one of the more abundant metals. And by tuning the exact crystal structures, we can synthesize materials that evolve oxygen really quickly. So you can see these are bubbles of oxygen. And when we think of, and the reason I brought this picture is because I know lots of people here are working on chemical reaction networks. And you know, in the later parts of this talk, I will also talk about chemical reaction networks. But like in the real physical system, this is kind of what, especially when, in my case, when I'm trying to develop theories to explain catalysis, this is actually the system that I'm tackling. And although, for example, maybe the reaction wouldn't go as fast in, for example, the origin of life or something. But anyway, when I start thinking about, when I see these pictures, I was hoping they would be kind of stimulating to some people. So, because I think in the general chemical reaction network picture, then, for example, we don't consider things like, does the surface area change? Or like we assume CSTR, but I mean, in this case, it's very, very non-homogeneous. And so I thought this would be interesting for some people here. Anyway, based on some kind of material synthesis, we can synthesize good materials. And I mean, lots of people around the world are trying to do that. And one of the key criteria of making better materials is increasing the lifetime. Meaning, of course, we want the reaction to go faster. We want the reactor to produce hydrogen faster. But for example, if you, I mean, think about your phone battery. Like even if it has like a, I don't know, like even if it's super, display is super beautiful or something, if it only lasts 2 hours, you wouldn't buy that phone. And so the battery lifetime is a super important component of smartphone. And also in the electrolyzer, the lifetime of the device is super important. And by lifetime, I mean, people typically expect like 3 to 5 years. And what that means is, of course, how fast the reaction can proceed is important, but also how long can that reaction be sustained is also important. And I'm hoping you would kind of see hints of, you know, connection to the origin of life, because that is also how long can you sustain some chemical reaction kind of question. So, of course, when you in the experiments, of course, we can measure the lifetime. And measuring is easy, except it takes a really long time. Of course, if you want to measure a 3-year lifetime, it takes 3 years. Even if you do some kind of extrapolation from, you know, like a cheaper timescale experiments, it still takes, for example, 1 year or several months. And the alternative approach that is used by the mainstream of the chemistry society is DFT. DFT stands for density functional theory. It's essentially you're solving the Schrödinger's equation numerically, and then based on some atomic composition, you calculate the properties of the material. And then you can see, okay, this composition is stable, this composition is not stable, and so on. And so if you have a stable composition, you can expect it to have a longer lifetime. And if it's an unstable composition, then you can expect it to have a shorter lifetime. The problem is, this is equilibrium thermodynamics, not the non-equilibrium statistical thermodynamics. That it's the, you know, standard textbook old thermodynamics. And what that means is it gives you a binary answer whether it's stable or not. So stable in this sense means stable forever, like a timescale, infinity timescale stable. And of course, in reality, almost no materials are like that. And even if you do have such a material, maybe it's not active. And actually, in industry, a lot of the materials that we use are called metastable. Metastable means not thermodynamically stable, but they are stable enough that, for example, by the time they die, we don't care. So, for example, if you require a time scale of 5 years, maybe metastable materials have a time scale of 10 years, and that's enough for our application. And so these kind of thermodynamic approaches, they skip over all of those metastable materials. They just predict them as not stable. And I mean, theoretically it's true, but in terms of looking for materials, that's not the answer we're looking for. And so one of the main targets I'm working on is, can we predict the catalytic lifetime from, for example, accelerated aging test or some different experimental data? And this is why I'm kind of working on trying to make a theory of catalysis. And the conventional approach in catalysis is to start from a well-defined chemical reaction network. I mean, you wouldn't. I mean, strictly speaking, a chemical reaction network. If you define some species and reactions, I mean, you could define you could call it a chemical reaction network. But I mean, this is a super simple chemical reaction network. So explicitly, this asterisk stands for a open site. So a vacant site, for example, a metal atom on the surface of a solid material. And then you could attach, for example, a proton and an electron, and you could make hydrogen, adsorbed hydrogen. This ADS stands for adsorbed, so it's on the surface of the metal. And then you could attach another proton and an electron, and then you can convert that to hydrogen. And this is being super explicit in all of the components, but in terms of, for example, Feinberg notation, then you could just ignore all of those H plus and E electrons and hydrogen. You could consider them buffered, or there's enough of them, or their concentrations don't change or something. And then you can simplify the network, and you can call it a two-state model. So there's a site that's open and a site that's occupied. And the conventional approach in catalysis is you start with such a well-defined mechanism, and then you apply the steady state approach. So, for example, you would set you could set the adsorbed site, and you could calculate how it changes with how the amount changes with time, and you could set this to 0, and then you can calculate the reaction rate from this. So for people that are familiar with biochemistry, you could consider this very similar to the Michaelis-Menten. So Michaelis-Menten also you have a E plus S goes to ES. So one of those enzymes is not occupied, and one of them is occupied, ES state. And then you would apply the steady state to the occupied state. I mean, mathematically, it doesn't matter which one you apply to steady state because the total amount is conserved. But anyway, the approach used in biochemistry and how to say the chemistry is quite similar. You would apply the steady state approach to a specific reaction mechanism. And what people would do, for example, this is one of my previous work, is you would compare the results with experiment. Like, for example, this black one is experiment. It's the amount of, the y-axis is the current density, which is the amount, corresponds to the amount of hydrogen generated from the catalyst. The x-axis is how much the electrode potential, which means how much electrons we're force feeding the electrode. And so as we force feed the more electrons to the electrode, it generates more hydrogen. And in the case of the original mechanism that I showed you on the previous slide, I mean, even if I try to fit the parameters better, it doesn't work. But then if you add an additional reaction, then it fits better. And so this is kind of the approach that people would use to see if, number one, their mechanism is correct. And then they would try to, for example, guess parameters and try to think, okay, which kind of parameters would make this even better, and so on. But then I'm sure the people here are thinking, okay, but what if we don't know the mechanism? Like, this is one of the central questions, for example, in, I think, for people working with metabolism, then, like, well, I shouldn't say no way, but at least we have a list of known chemical reactions, but that doesn't mean the database is complete. And that doesn't mean we can detect all of the reactions. I mean, even detecting all the species is difficult. And then it's even more difficult to detect what each of the species are doing. So what if we don't know the mechanism? Then, as I showed you before, then we typically people would, how to say, alter the mechanism so that it fits reality even better. But then you could, you can imagine this is just you can continue this forever. And also, what if the steady state approximation can't be used? What I mean is, like typically in the time scale of experiments, for example, you have time and then you have activity, and the catalyst dies after a while. So until it dies, when it dies, the activity drops to 0. And until it dies, you could consider this to be kind of like a quasi steady state. So it's pretty much constant. At least in the time scale of experiments, for example, this would be like, this entire x-axis would be like, I don't know, months or weeks. And then the time scale for performing one reaction, generating one molecule of hydrogen, that would be in the time scale, for example, seconds or minutes. So, I mean, you could consider there's a quasi steady state here. And then the true steady state, in this case, it would be chemical equilibrium. But then the true steady state is after the catalyst dies and nothing occurs anymore. But then if you want to know the lifetime, it's not the steady state. And so if you want to look for stable materials, or like if you want to predict what is the lifetime of this chemical reaction system, then the standard steady state approximation where you apply the steady state to everything, it doesn't work. And so based on this kind of thinking, this is why I started thinking about timescale decomposition, which is some of those processes in the catalysis are fast. Namely, the main reaction, for example, generating hydrogen, is fast. And then the catalyst deactivates over time in a timescale of, for example, months or weeks or months. And at least in our systems, that happens by the metal oxide dissolving into the solution. And so that is a process that is, for example, 100 or 1,000 times slower than the main catalytic reaction. So the main catalytic reaction here is shown in blue. This is maybe 2 or 3 orders of magnitude faster than this red one. And so that means maybe we can separate the time scale, meaning we can approximate this one to be in the steady state relative to this red deactivation process. And then in that case, we can define an effective rate for the deactivation, which is given by this equation here. And in the next slide, in the following slides, I want to show you kind of an overview of what we can do, how I got this, and then what we can do with this. So this is the same network that I showed you in the previous slide. And then the ODE for this one is in this form. So C is, you could, I mean, originally I considered this to be catalysis, but you can consider this the conserved part. Both are fine. So the conserved part, there's one cycle. Everything is mass action. It's single particle mass action. So if you look at the columns, all of them are the summation of along each column is 0. So everything is conserved in this network, in this blue part. And then the dissipation, the dissolution happens from these red reactions. And these are also single particle mass action in this model, meaning all of these coefficients, they're diagonal, they're along the diagonal. So we have a conserved, you could call this a weighted out degree Laplacian, graph Laplacian. And then this is a diagonal matrix. So then this setting makes it easy to calculate things. So actually, if you apply the steady state approximation to this blue part, so you set Cx equals 0. Sorry, I should have mentioned. I mean, for most people, I think it would be maybe obvious, but x is the vector of the concentration for x1, x2, and so on. So then eventually over time, all of the x's, this is it goes to, all of the x's go to 0. And the time scale which x goes to 0, this is the time scale I'm calling the lifetime. And mathematically, I define that by saying when the total amount of species decreases to 1 over e of the initial value. So applying the steady state approximation to this blue part, calling the total number of x's n initially, or n at that time point, and then we have the rate constant. And then if you set each xi like this, then it will satisfy the steady state approximation. This is easy to verify. And so you can calculate how n should change with time. And because Cx is 0, so the only process that changes the total is dx. And then you can calculate this. And so we have the theoretical equation for the lifetime that I showed you on the previous slide. And we can, because I belong to an experimental lab now, so it's we can easily check whether this is correct or not, or how accurate it is. So we have a electrode, we have a catalyst on this side, and then we have a we take a UV vis spectrum every 15 minutes in the reactor. And what this UV vis spectrum is doing is it's measuring the amount of chemical species dissolved into the reactor. So on the right side, you see one representative example. Each of these are a UV vis spectrum taken every 15 minutes for 60 hours. And then at each time point, we get a spectrum, and then we can fit the spectrum with the reference spectra of different species. So this red one corresponds to manganese 7+, and this blue one corresponds to manganese 3+. So these are the species that we detect in the solution. So the gamma manganese oxide, this is a specific type of manganese oxide solid material. And then over time, it dissolves completely into the electrolyte chamber. And then because it completely dissolves, so then eventually the activity decays towards 0. So this black one is the current density, and after around 40 hours, it decays to 0. And this red one starts increasing from the initial time point. That is the manganese 7+ that is dissolved into the solution because the manganese oxide is dissolving. And you can see at some point, we're not exactly sure why it shows such a sharp peak here. But anyway, around the time the catalyst dies, also it seems the dissolved species seems to switch from manganese 7+ to manganese 3+. So something is going on here. We don't know exactly what, but anyway, this is the kind of experiments we can do. And so we can apply the theoretical equation of lifetime into this manganese oxide network. So in this case, we based on previous studies, we kind of know the mechanism. Know meaning we know it passes through these manganese 4+, 2+, 3+ intermediate states. And also we know this process is very slow. So RDS stands for rate determining step. And then, so because this is the rate determining step, we know this process must be fast. And then we have a rate constant here for dissolution that we can measure. And so the experimental lifetime is on the on the y-axis, and then on the x-axis we have this VD, because we don't know the exact rate constant for the dissolution. We just know the how fast it's dissolving. So this is the rate of dissolution, not the rate constant. It's similar but not exactly the same. This JOER, this catalytic reaction is called OER, oxygen evolution reaction, and so J is the common notation for the current density. So essentially this is saying how fast is catalysis happening. And so here we assume this is the proxy for the rate constant of the rate determining step. So ideally we would have liked to put log k RDS here, but because we couldn't get the exact rate constant, so it's log JOER. So the units on the x and y axis are not exactly the same, which is why there's no unit on the x axis. But anyway, you can see a kind of a qualitative trend saying that it's the theoretical lifetime and the experimental lifetime seem to match kind of okay-ish. And so even for some kind of like a realistic catalysis system, this kind of gave me support that maybe this timescale decomposition is working. And so recently I'm thinking of applying this to the origin of life and and especially for autocatalytic networks. So let me explain a little bit about my kind of speculation. So in the case of the origin of life, I'm kind of imagining a network of course there should be some kind of proto-metabolic reaction. And then if you think about an open system, then each of these species has a chance to diffuse away. And there might be some external supply coming from, for example, hydrothermal vent. It doesn't have to be, but anyway, some kind of supply. And then for this kind of network to leave the supply, in the vicinity of this supply, then you could kind of imagine the supply and diffusion kind of balancing each other out. But then I mean, eventually this kind of network has to go away from this supply. And then in that case, you would require some kind of process to increase the amount of chemical species, for example, autocatalysis. So the ODE that we can set for this system is very similar to the one that I set for catalysis. So we have a conserved part. So this is the, I put this L for Laplacian. But anyway, this is the conserved part. And then we have something that corresponds to autocatalysis and diffusion. And both of these, I'm still assuming single particle mass action. So in this case, they're both diagonal matrices. But anyway, because they're both diagonal matrices, so I mean, we can just call their sum delta. And then just applying the same timescale decomposition, we can calculate the eigenvalue of this network. And then based on this network, based on this eigenvalue, for example, if the largest eigenvalue is negative, then we know that the network is going to dissipate. And then if it's 0, then it's stable. And then if it's positive, then we can expect exponential growth. So, I mean, let me show you some examples. So, for example, we can set some, I don't know if you call these basis vectors. I mean, maybe you could call them basis matrices, I guess. So, for example, you could set a base matrix for each of these. For example, the Laplacian part, you could set this one. And for the dissipation part, maybe you could set this one. And for the amplification part, autocatalysis part, maybe you could put one of the species has an autocatalytic amplification. And then each of these, you could consider them to be just the stoichiometry, not the rate constant. And then I can multiply them by, for example, some factor to change them by a rate constant. For example, instead of 1, 0, 0, maybe I could have like a 0, 0. And so what I'm putting here on the x and y axis is whatever, I'm multiplying this Laplacian by some constant factor L, and then I'm multiplying this autocatalytic matrix with some constant factor A, and then I'm multiplying this one by D, and then I can calculate the eigenvalue. And then in some areas, the eigenvalues are negative, and in some areas, the eigenvalues are positive. And so along this white line, this is where the eigenvalue is 0, and this is where it's stable. And then where the eigenvalues are negative, this leads to the chemical reaction that we're completely dissipating, and that would lead to extinction. And then you would have exponential growth on this top part. So where the amplification is, the factor of amplification is very large, then you would have exponential growth. And what I think is interesting to share about this is I mean, this is just based on intuition, but autocatalysis is not common compared to diffusion, at least. All of the chemical species can diffuse away, but autocatalysis requires a very specific mechanism. I mean, if you ask a chemist, please generate an autocatalytic network, it's not easy. So in this case, I put all of the chemical species having the possibility to diffuse away, but only one of the species having the possibility to be autocatalytic. And because of this imbalance, this boundary is not a straight line between a diagonal straight line. It's more curved upwards, meaning the region for exponential growth is that much smaller. And that's because it's much easier to just diffuse away. And so, I mean, maybe this is intuitive, but I mean, I see diffusion as a key obstacle for proto-metabolism. And so one of the ideas would be, for example, if you have higher catalysis for autocatalysis, then you could increase along the y-axis. And then if you have some kind of membrane, then you would be lucky and you would, suppress diffusion. And then that would bring you into this region. And finally, this is it's just numerical simulations, but I thought this would be interesting, so I wanted to share it with you. So because these are numerical simulations, so even if it's something that we can't get the analytical solution, numerical simulations are easy to do. And so I was wondering, what if the environment fluctuates? So environment in this case, meaning the S vector, the supply vector, we can allow it to fluctuate. And so at some time it's off, and at some time it's on meaning there is a supply, and then it switches on and off again. And you can consider the possible metabolic response. These are just possibilities, but you can consider one where it just ignores the fluctuation and it doesn't respond at all. You could also consider a situation where the metabolic response is in phase to this one, meaning autocatalysis is turned on when there's a large flux, and autocatalysis is turned off when the environment supply is gone. And also, you could imagine an opposite scenario where you save for a rainy day. So you turn on autocatalysis as soon as the supply is gone, and then when the supply is gone, you become lazy and you turn off autocatalysis. So I mean, there are several possibilities of metabolic responses, and then based on numerical simulations, we can see which one is better. And so I mean, these are numerical simulations, but then if you have in phase, it grows much faster. And to me, what that seems is, I mean, of course, I don't know how in phase or out phase can happen, but then if there is a choice, if, for example, we have two networks or two systems, one of which is in phase and one of which is out phase, then this in phase one is going to completely dominate over the out phase because the out phase grows much slower. So I don't really know why this is not better or why one is better than the other, but then this is kind of a evolutionary, I mean, this might be some kind of evolutionary selection. If you can do signal processing, if you can detect when the supply is here and you can turn on autocatalysis at that point, then nature will select that network. And yeah, that is the end of my talk. So I want to thank my collaborators in my lab, and also I want to thank my collaborators, mostly applied mathematicians and physicists, for their advice in doing this kind of work. And thank you everyone for your kind attention, and yeah, thank you.